Electroporation uses an electrical pulse to create temporary nanopores in the lipid bilayer of the cell membrane. This increases the permeability of the membrane and transports macromolecules into the cell. Electroporation can be used to introduce chemicals, drugs, and DNA into the cell for the purpose of investigating cellular morphology, development, and function in vivo. This video is a step-by-step guide for electroporation in tadpoles that can be optimized for species-specific and tissue-specific applications. Pull glass capillaries using a pipette puller and break the tip at an angle using forceps to create a beveled tip.
That's it.
Backfill the pipette with mineral oil, making sure that there are no air bubbles. Put the pipette onto the plunger and tighten. Familiarize yourself with the control knobs on the micromanipulator. They can move the device in the X, Y, and Z direction. Adjust the injection volume to your desired amount by changing the switches on the side of the nanoject. Press and hold the empty button on the nanoject to remove enough mineral oil to load 1 to 2 microliters of solution. To fill the pipette, we recommend placing a droplet of solution mixed with 0.01% fast green onto a piece of parafilm, then lowering the tip of the pipette into it. Press and hold the fill button on the nanoject until a sufficient amount of solution is loaded. The ideal electrode setup is dependent on the tissue that is being targeted. For this demonstration, we will be electroporating the brain. We ran each wire of the electrode through a plastic tube mounted on a micromanipulator and fixed the needles such that they were in position to make contact with either side of the brain. Clamp the exposed ends of each electrode wire to the capacitor. Clamp an additional 2 wires on either side of the capacitor. Then wrap the end of one wire around the plus knob and the other around the minus knob on the stimulator. Adjust the settings of the stimulator to fit environmental parameters using the knobs. We anesthetize tadpoles by bathing them in 0.03% MS-222 buffered with 0.1% sodium bicarbonate prior to the electroporation procedure. The amount of time it takes for the anesthetic to take full effect varies by individual, so we recommend periodically checking movement in response to stimuli to confirm that the tadpole is fully sedated before continuing to injection. Carefully move the tadpole from the anesthetic to the injection setup. The way you position the tadpole will depend on what tissue is being targeted. Slowly lower the pipette and adjust its position as needed. It's okay if the tadpole flattens in response to the pressure of the pipette, but be careful not to push through the target tissue into a different region of the body. Once the pipette punctures the correct tissue, press the inject button on the nanoject. We recommend waiting 5 to 10 seconds between each injection. Following injection, orient the tadpole's head towards the electrode. Lower the electrode until it is in full contact with the tadpole. Push the mode switch to single to deliver N=1 pulse. The tadpole will twitch in response to the pulse if it was successfully received. If you observe excessive skin burning as a result of the electroporation, try a new electrode or lower the pulse number and/or voltage. Following electroporation, tadpoles are transferred to fresh water for several hours to recover. We recommend checking tadpoles hourly to observe behavior and identify deceased individuals to quantify mortality. Electroporation can be used for a variety of applications, including visualizing cells, introducing foreign genes, and manipulating gene expression. To date, electroporation protocols are largely limited to model species. Developing protocols beyond Xenopus offers the opportunity to explore questions related to organismal biology, ecological and evolutionary biology, and neuroethology across a wider diversity of amphibians.